Was it just me, or does today seem like it's just so many more things to do today than you would normally have? Is it just me, or is it? I see a lot of people shaking their heads. I feel like I've just been going like on a treadmill the whole day long, and it's like maybe I shouldn't have had a 3-day weekend. I don't know what happened. Something happened. Okay. Well, I guess we'll continue that tradition with my lecture. So, putting you on the treadmills, as it were. So the exam is graded. I will give the exam back to you at the end of the period. And the average on the exam was 68. I haven't drawn up the overall curve yet. It was lower than last time, but 68 is not a bad score. We are rapidly moving towards the final. And I think I told you on the final, you will get to use a note card. You have to get the note card from me. I will bring those in next week. So make sure. And you have to have a note card from me. If you don't have a note card from me, you will lose points. So even if you don't want to use a note card, you have to have a note card from me. That's the price of admission. Okay? So we'll talk about that next week. Okay. Today what I'm going to do is I'm going to go through and finish up this little section here on the glyoxylate cycle. And then we'll turn our attention to electron transport and oxidative phosphorylation, which is where the vast majority of the energy is made in the cell. Okay? So the glyoxylate cycle is actually a cycle that is very closely related to the citric acid cycle. It uses several of the enzymes of the citric acid cycle, but with some important differences, and I'll show you those. So let's take a look at the cycle first. This is sort of what it looks like. Okay? Now, let me tell you a little bit about the cycle before I show you the reactions or tell you about the reactions inside of it. The glyoxylate cycle is a cycle that does not run in human beings. It does not run in animals. Okay? It runs in plants, bacteria, yeast. They can run the citric acid cycle. We cannot do that. And that's because they have two enzymes that we don't have. All right? They have this enzyme here called isocitrate lyase, which is one that's not found in the citric acid cycle. And yes, I think that's a good enzyme name to know. And they also have an enzyme called malate synthase, which we don't have. And yes, that's also a good enzyme to know. All right? Now, because they have these two enzymes, they can short-circuit the citric acid cycle. That's really what they're doing. And in short-circuiting the citric acid cycle, they have some real advantage. All right? Now, I'll tell you about that in just a second. So, the glyoxylate cycle starts out just like our citric acid cycle starts out. You combine acetyl-CoA with oxaloacetate, and you get citric acid, or citrate. Citrate gets isomerized into isocitrate, just like in our cells. And it's the same enzymes. And by the way, this cycle occurs in addition to the citric acid cycle in these organisms. So they can run both of them, they can run one of them, they can run the other one. It doesn't really matter, but they've got both cycles that they can run. And depending upon the circumstances, they'll run one versus the other. We'll say a little bit more about that later. Okay? All right, so they get to isocitrate. In us, that occurs as well. And in us, at isocitrate, we still have 6 carbons. All right? The next step in our citric acid cycle, or in their citric acid cycle, would be to decarboxylate and go down to 5 carbons. However, the glyoxylate cycle does not do that. That's where the shortcut comes in. So instead of decarboxylating, the glyoxylate cycle splits this 6-carbon molecule into 2 pieces. One of the pieces is known as glyoxalate. That's where the cycle gets its name. Okay? Glyoxalate can combine with a second acetyl-CoA, because remember we started with 1 up here. It can combine with the second acetyl-CoA. That makes malate. And then malate can become oxaloacetate. All right? So we've short-circuited the cycle, and what we've done is we've taken a 2-carbon piece off of this, combined it with another 2-carbon piece, and made a 4-carbon piece. And if you're sitting here thinking about this, you're thinking, well, Kevin, what about the other 4-carbon piece that was left? Well, when you split isocitrate into glyoxalate and something else. The something else is succinate. So this succinate, of course, goes through the regular reactions of the citric acid cycle. Succinate to fumarate to malate. Well, now we get over here, we've got 2 malates, and we get up to here, we've got 2 oxaloacetates. We started with 1, we now have 2. That's a net gain of 1. And we don't need 2 to make the cycle go, we only need 1 to make the cycle go. This other one can be used for something else. One of the things it can be used for is to make glucose. Now, this was the cycle I mentioned to you the other day, that if we had it, we would have a nice, dandy way of burning up fat, because acetyl-CoA we can get from fat quite readily in addition to getting it from glucose. So if we wanted to burn up a whole bunch of fat, this would be a really nice way to do it, and then convert it back into glucose. Converting fat to glucose would be nice because we can get rid of glucose pretty easily. Unfortunately, we can't do that. All right? Now, well, when does it do the citric acid cycle, when does it do the glyoxylate cycle? That's actually kind of a complicated question. And I'm going to give you an answer, but I'm not going to really expect that you're going to be able to tell me when one versus the other goes, which basically means you're going to turn your minds off at this point, I imagine. But I'll tell you anyway. Okay? This cycle doesn't have that decarboxylation. This cycle doesn't have the second decarboxylation. And both of those decarboxylations gave NADH. This cycle gives 1 FADH2, and it gives 1 NADH for each one of these guys going through. So in essence, we get 2 NADHs. But that's still less than what the citric acid cycle would give. That means, therefore, that when the cells have plenty of energy, they're not going to be running the citric acid cycle. They're going to be running this cycle and taking that extra oxaloacetate and making either glucose or aspartic acid. And when they're low on energy, they're not going to run this cycle. They're going to run the citric acid cycle. That's basically what it comes down to. Now, when we talk about photosynthesis later, we'll see how glyoxylate plays into this. And we will also see why it's important that plants have this cycle. Okay? So, just keep that planted in your head. We will talk about glyoxylate again later. Okay, questions about the cycle. 2 enzymes, 2 unique enzymes make it possible. Everybody's too tired to ask a question. Yes? Oh, okay. So succinate's 4 carbons, glyoxylate is 2 carbons. Okay? So we take 2 here, we add 2 with acetyl-CoA, and we've got malate. Okay. Well, that is the glyoxylate cycle. Now we turn our attention to the most important energetic considerations, certainly in animal cells, okay, and in bacterial cells. Plants, of course, have photosynthesis, and they've got other energy sources. But for us, this is pretty important because this is where we're probably going to get 95 or more % of our energy. All right? It involves 2 processes. It involves electron transport, and it involves oxidative phosphorylation. These 2 processes normally are what we call coupled. They're coupled, meaning that one depends on the other. We could think of them as like codependent people, right? They are codependent. They rely on each other. If oxidative phosphorylation is not running, electron transport will stop. If electron transport is not running, oxidative phosphorylation will stop if the 2 processes are coupled. We will see that one can uncouple them, and we'll see some interesting things with that. But if they're coupled, they're totally dependent upon each other. Okay, well, let's talk about electron transport first. I've already talked about mitochondrial membrane or mitochondrial structure. I'm just going to remind you of what I showed you earlier. I told you at the time that the most important component of the mitochondrion is the inner mitochondrial membrane. And it's particularly important for electron transport because the reactions of electron transport mostly occur in that membrane. They mostly occur in that membrane. Okay? We'll see that there are complexes that handle the electrons, and they're embedded in the membrane. At least 3 big complexes are. Okay? Actually 4. All right. There's the matrix. That's what I described as the sort of cytoplasm of the mitochondrion. That's the sort of soup of everything. And then there's the outer membrane, and finally there's the intermembrane space, the space between the inner and the outer membrane. Okay? The inner membrane has a very important property, and it's essential for electron transport. The property that it has is it is very, very impermeable to things. Okay? So we talked earlier about how membranes let only a few things go through readily. Anybody remember what they were? Water, CO2, carbon monoxide, and oxygen. 4 things, okay? Those can pass through the inner mitochondrial membrane as well. And that's good because oxygen has to get inside of there, okay? But what cannot pass through there is something as tiny as a proton. Protons cannot cross the membrane without assistance. And that turns out to be key to how oxidative phosphorylation, the next process, works. So the inner mitochondrial membrane is impermeable to protons. It's impermeable to protons. Okay. When we look at electron transport, we see that electron transport is taking electrons away from electron carriers. So we've talked about 2 main electron carriers so far, NADH and FADH2. Okay? And I told you how those were in short supply in the cell. If we don't offload those electrons and protons off of those carriers, then the cell will have no NAD and no FAD, and then it can't do any oxidation. All right? So it's important that those get recycled. And the way in which those electrons and protons from NADH and FADH2 get recycled is by the electron transport system. So these complexes dump electrons and protons into these carriers. Okay? Now I'm going to simplify this for you in a minute. So this looks a little hairy. It's not too hairy. Okay? NADH dumps its electrons into a carrier that we call, well, let's see. Maybe we can find an easier way to describe it. No, I don't think so. Well, it turns out that the easier thing actually is on here. All right? NADH dumps its electrons into complex 1. So if we follow the path of electrons, NADH dumps its electrons into complex 1. The protons go separately, so we don't have to worry about the protons. All right? So all we're going to follow here are the electrons. Complex 1 gets the electrons, and then it passes the electrons onto something called coenzyme Q. That's CoQ right there. And then coenzyme Q takes and dumps its electrons into this monstrosity over here that we'll call complex 3. So from NADH to complex 1 to coenzyme Q to complex 3. And then complex 3 dumps its electrons into this thing over here called cytochrome C. And then cytochrome C dumps its electron into this complex 4. And then last but not least, those electrons have to go somewhere. Complex 4 dumps those electrons onto oxygen, which grabs a couple protons out of the solution and makes water. Now, the reason when you're exercising that you start breathing so heavily is because this process is going on like crazy. And your body is using oxygen as a thing to dump the electrons into. You're making water in the process, but your body is needing oxygen, so you start breathing heavily. That's why you're breathing heavily, to make up the oxygen that's being used right here. Okay? Now, the one thing I didn't tell you about was complex 2. What's up here? What's that? Well, complex 2 is up here on the top, and complex 2 accepts electrons from FADH2. So electrons from FADH2 enter through complex 2. This complex 2 is embedded in the mitochondrial membrane, like essentially all these are. I'll point out the ones that aren't, but essentially all of them are. Okay? Complex 2 dumps its electrons into coenzyme Q, and coenzyme Q dumps them all the way through the same system you've already seen here. Okay. Now, that's the electron transport system. Yes, I think you should know the order of the carriers. I'm going to repeat them for you. NADH to complex 1, FADH2 to complex 2. Both of those converge on coenzyme Q, otherwise known as CoQ. Now, CoQ has an interesting property. CoQ accepts electrons in pairs. The electrons come in from NADH and from FADH2 in pairs. But all the other carriers only accept electrons one at a time. So coenzyme Q acts as what I call the traffic cop. It accepts them in pairs, but it passes them off one at a time. Okay? From coenzyme Q, electrons go to complex 3. Yes? So, I'm not sure I understand the question, but this enzyme right here turns out to be one of the enzymes of the citric acid cycle. So that's why that reaction is showing you that way. Okay? So it's the only enzyme, last time I mentioned, I'm not even sure if I brought it up after I mentioned it, but it's the only enzyme of the citric acid cycle that's not found in the matrix. It's embedded in the membrane, and it's the same as this enzyme right here. Okay? Okay. So electrons go from coenzyme Q to complex 3. They go from complex 3 to cytochrome C. And from cytochrome C, they go to complex 4. Complex 4, they go to oxygen, and they make water. That's the electron transport system. Now, there's a little bit more to it than that. Okay? But that's the order of events in which the electrons move through the cells. Okay? Well, there's something else that's going on here. If we simply had moving of electrons going on and nothing else, there wouldn't be any way to make ATP. Okay? This is a cute little electric circuit, as it were. And it's not unlike an electric circuit. But there wouldn't be any way to make ATP based on just the movement of electrons. The movement of electrons through the complexes, though, causes something to happen, and that something to happen is what makes possible the synthesis of ATP later. Okay? What's happening is that as electrons pass through complex 1, electrons are moving from left to right in the direction of coenzyme Q. As electrons are passing through complex 1, protons are being moved, or pumped, as I like to say. Protons are being pumped out of the matrix and into the intermembrane space. So protons are being pumped out of the matrix from complex 1 into the intermembrane space. What does that do? That causes a proton gradient. And what does a proton gradient do? Well, the protons want to come back in, and if we can find the right way to get them back in, we can harvest that energy, and that's what we do. I'll show you how that occurs later. But suffice it to say that by pumping protons out of the matrix and into the intermembrane space, we are doing the equivalent of what I like to think of as charging a battery. You charge a battery so it'll have juice later, and the juice is in the form of the electrical potential and the electrical potential is very real because you've got a charge difference across the membrane. More protons outside than inside. Complex 1 is not the only place where protons get pumped. In fact, wherever you see the word complex up here, that's a proton pumper. Complex 1 pumps protons, complex 3 pumps protons, and complex 4 pumps protons. And no, you're not going to have to draw all these different things up there. Okay? Keep it simple. All right. Now, what this means is that if electrons come in through FADH2, they miss complex 1. It means that electrons coming from FADH2 don't charge the battery as much. They don't charge the battery as much. Now, if you go read textbooks, including my own, all right, you go read textbooks, people get all arguing about exactly how many ATPs you make per NADH and exactly how many ATPs you make per FADH2. And the fact of the matter is, it really doesn't matter a lot. Okay? You make some. We're going to, in this class, say that you make 3 ATPs for every pair of electrons coming from NADH, and that's approximately right, and that's close enough. And we will say that there are approximately 2 ATPs made as a result of electrons coming from complex 2. Okay? Now, notice that we haven't made any ATPs up here yet. All we're doing is charging the battery so that later we can make those ATPs. But we're making enough charge that we can make 3 ATPs starting from the left side, 2 ATPs starting from FADH2 on the top. Everybody with me? Yes, question here. Pumping of protons occurs at complexes 1, 3, and 4. That's correct. Question? Yeah, no, a pair of electrons. So every NADH and every FADH2 has a pair of electrons that it donates to this process. So for each pair, we get approximately 3 ATPs from NADH and approximately 2 ATPs from each FADH2. Everybody with me? Okay, so that's what's happening in the process of electron transport. All right? Let's see, what was this? This is a nice scheme of it. Okay? This scheme shows, all right, here we are, down here, the bottom is the matrix. The top part is the intermembrane space. Here's the inner mitochondrial membrane that we can see. And what we see is that electrons are entering into complex 1. Complex 1 is a complex. It's big and hairy, and it's got several proteins in it. And electrons move through several things within it. Complex 1 dumps things off to coenzyme Q. Coenzyme Q dumps things off to complex 3. And complex 3 dumps things off to cytochrome C. And cytochrome C dumps things off to complex 4. At complex 4, we see oxygen being used and water being produced. This is not showing FADH2. But FADH2 would simply be coming in through here. Okay? Makes sense? Now, complexes 1, 3, and 4, as you can see, are pretty well embedded in the inner mitochondrial membrane. Coenzyme Q is sort of peripheral, and cytochrome C is sort of peripheral. This figure doesn't show it that much. Okay? But they're not nearly as embedded as these big complexes are. Okay. Now this process that you see on the screen is interesting because it's conserved across almost all living cells. This is a very, very efficient way to make ATP. Bacteria have versions of this in their membranes just like we do. Okay? Bacteria don't have mitochondria, but they use their whole cell like it's a mitochondrion. Okay? They use their whole cell like it's a mitochondrion. Okay. All right, so that's electron transport. I'll just show you this. You're not responsible for it. But I told you that coenzyme Q was the traffic cop. Coenzyme Q is capable of taking 2 electrons and passing them off one at a time. Here it is with 2 electrons, and here it is passing them off one at a time, ultimately getting up here to the oxidized form where it's passed both of the electrons off. You don't need to know that, or you don't need to draw that, but you should certainly know it's the traffic cop, because I think that's an important component of that. All righty. It's Tuesday. I think we should stretch or something, right? No? Oh, uh-huh. It makes a difference. You know, it really does make a difference when you're sitting here and you're kind of bored, just kind of like get up and get the oxygen, get the stuff flowing. Okay. I will tell you the cutest joke I saw on the extra credit from Friday. It was my favorite, and it went as follows. Why was the little cookie crying? Because his mother was away for so long. His mother was away for so long. Cute, cute. Okay. All right. That was my favorite joke. Well, we're talking about a subject. Yes. Yes. Question, how many hydrogens are moved along at each point? People spend their whole careers arguing about that, and I don't. So there are a few. The estimates are that something like about a dozen or so total, a dozen to 16 may get moved across for the entire process. For the entire process. So for the movement of one pair of electrons all the way through the system, about 12 to 16 protons. Where is it picking the protons up? Remember, water has a pH of 7, which means it has a concentration of protons of 10 to the minus 7th molar. That's where it's getting them from. Yep. If you have an aqueous solution, you have protons. No ifs, ands, ors, or buts about it. But it's a good question. It's a very common question also. Okay, so there's plenty of protons to move across. All right, oxidative phosphorylation. So we have charged the battery. Now we're going to discharge the battery, and we are going to make ATP in the process. Okay, so battery is one analogy that we can give for this. I think perhaps a better analogy than a battery, though, is pushing water uphill. Okay, if I were to take all of the rainfall that happened yesterday in the Willamette Valley, and I were to go and collect all of that and put it up on top of Mary's Peak, okay, I would be doing something equivalent to charging a battery, because I would be creating potential energy. I'm putting that water up at a higher altitude so that when it flows back down, it could turn a turbine, and that turbine that it turns could, in fact, generate electricity. Okay? So that's what the whole idea of hydroelectric power is and how it works. Well, that turbine is not exactly a crazy thing, because it turns out that ATP synthase is the way in which protons actually enter back into the matrix. I need to tell you a little bit about what's going on. In the process of oxidative phosphorylation, it occurs through something we call complex 5. Now, complex 5 has nothing to do with electron transport. It's one of the biggest things of confusion that students have. When I say complex 5, it has nothing to do with electron transport. Okay? There are no electrons that are moving through complex 5. Complex 5 is also called ATP synthase, S-Y-N-T-H-A-S-E. And this is what it looks like. This is what it looks like. Now, this is a little upside down from what we saw before. The intermembrane space here is on the bottom. The matrix is on the top. But this thing is really remarkable what it does. It's one of the most remarkable machines, and it literally is a machine that's in your body. Okay? You see these little chambers right here, these little blue guys here, and the little green things that go with them. These are chambers where protons are moving. Remember the proton concentration as a result of electron transport, there's more protons out here than there are in here, and the protons want to come in. They can't short circuit the membrane because the membrane won't let them short circuit it. Instead, they have to come in through these channels. And when they come in through these channels, something really remarkable happens. This turbine down here spins. It literally spins. And the output of that spin is ATP. The result of the spinning of the turbine, as we will see, is ATP. Now, this is absolutely remarkable. And I have a figure, if I can pull it up here, to actually show you the thing in process. Here's the cool video. You can watch this on your own as well. But let me just show you this. If it comes up, please come up. Oh, they're going to have a little commercial for us. But it's worth it. Intermembrane space up here, matrix down here. Here's the protons coming in.
The flow of protons down the electrochemical gradient drives a rotor that lies in the membrane. It is thought that protons flow through an entry open to one side of the membrane and bind to rotor subunits. Only permeated subunits can then rotate into the membrane, away from the static channel of some.
See the protons coming through, now they're coming back out in the bottom.
Subunits have completed an almost full circle and have returned to the static subunit. An exit channel allows them to lead to the other side of the membrane. In this way, the energy stored in the proton gradient is converted into mechanical rotational energy. The rotational energy is transmitted via a shaft attached to the rotor that penetrates deep into the center of the characteristic lollipop head, the F1 ATPs, which catalyzes the formation of ATP. The F1 ATPase portion of ATP synthase has been crystallized.
That means you can tell the structure when you crystallize it.
Its molecular structure shows that the position of the central shaft influences the conformation and arrangement of the surrounding subunits. It is these changes that drive the synthesis of ATP from ADP. In this animated model, different conformational states are lined up as a temporal sequence as they would occur during rotation of the central shaft.
Notice how it's being made in 3 different areas.
Like any enzyme, ATP synthase can work in either direction. If the concentration of ATP is high and the proton gradient low, ATP synthase will run in reverse, hydrolyzing ATP as it pumps protons across the membrane.
Pretty cool. Okay, so let me fill you in a little bit on what you've just seen. So, as I said, it's a remarkable machine, and it turns as a result of protons flowing through. Now, that's upside down again compared to that figure, but here's the intermembrane space. Here is the matrix up here. Okay. This structure has two components that we call F0 and F1. I like to think of it as a mushroom. Here's the head of the mushroom, and here's the roots of the mushroom. The roots of the mushroom are the F0. They're embedded in the intermembrane space. The head is where the ATP is made, and that's called F1. Okay? There are three places where ATP is made, and we'll see that there's a unit called beta and another called alpha, and it's actually these guys coming together that squeezes together the ADP and the phosphate. So you saw how that thing spun, and they told you that the structure of those proteins changed slightly. And so we've seen before how small structural changes can have big effects. In this case, the small structural change is to squeeze together phosphate with ADP, and that's what makes ATP. Okay? There are three of these guys. If we look at this from the top down, okay, so, or maybe the bottom up, I guess, is the way it would be there, but we see that three places where ATP can be made. All right? Three places where ATP can be made. Each of these places has a unique configuration at any given time. All right? They're called L, T, and O. L stands for loose. T stands for tight, and O stands for open. Now, let's watch and see what happens as we go through this, okay? Here is a loose sequence. Here's a loose structure right here. The loose structure is able to bind ADP and phosphate. All right? The rotor turns. We're still looking at the same structure, but now because that rotor inside of it has turned, it has changed the shape of the L into the T. So this L structure has changed into a T, and the T stands for tight, meaning that it's squeezing together the ADP and the phosphate. ATP is being made at this point. And you see that right here. When the rotor turns one more time, this T will turn into an O. Okay? This T will turn into an O, and what does O do? O lets go of the ATP. So we have three structures. Any one can be in any given state at any given time, and it sequentially will go from loose to tight to open. Loose to tight to open. Loose is where it will bind the substrates, the ADP and the phosphate. Tight is where it will squeeze them together and make ATP, and open is where it will release ATP. And that's the process. Yes? This is just sitting fixed, but the rotator inside of it is flipping those. Yeah. That's correct. Yes? You cannot make ATP from AMP in this thing. No. No. So you need a different enzyme to do that. Yeah. So this is called, well, this is actually, there's several things. So this is the F1 portion of the ATP synthase. And the ATP synthase was also called the complex 5. I can't pull it out. Complex 5. Thank you. But this is the F1 portion of that. Okay, yes? So when it runs in reverse, it'll just spin the other way. That's all it'll do. Yeah, it doesn't run in reverse much. That's not a common thing. They only told you about that so they could show you that spinning thing. So in a laboratory, they can run it in reverse. In your mitochondria, it's not doing it in reverse much at all. Yes? That is a typo. You are correct. Yes. Okay. This should be O right here. This should be an O. Okay. Now, why is this important? Well, this is really important because I told you that these two processes were dependent upon each other. They're codependent. Electron transport and oxidative phosphorylation. What did electron transport do? Electron transport created the proton gradient. What did oxidative phosphorylation do? It allowed the gradient to run back down. That's important because if you don't run the gradient down, then what happens is the gradient gets so high that electron transport cannot pump any more protons. It'll get stuck. Because you try to put too much water up over your head by pumping water and pushing water, eventually it's going to get so heavy you can't put any more up there. What oxidative phosphorylation